So, welcome back to Unit 3, Biological Basis of Behavior, Module number 11, Studying the Brain and Older Brain Structures. This is not that long of a module with not too many learning targets. So the learning targets, there are 3 of them for this module, are to describe several techniques for studying the brain's connections to behavior and mind. So like understanding the different techniques that we're utilizing to study living brains. The second learning target is to describe the components of the brainstem and summarize the functions of the brainstem, thalamus, reticular formation, and cerebellum. And then finally, to be able to describe the limbic system structure and function. So we're moving into our study of these neuroimaging techniques and an overview of the limbic system and the older areas of the brain. So I want to emphasize that what's really interesting about these brain neuroimaging techniques is that these ideas have really, what we've learned from neuroscience has really helped to debunk some many previously believed ideas like the 10% brain myth, phrenology, and brain size being related to intelligence, among many others. You just need to be thinking about how the ability to see the inner workings of the brain in action has really revolutionized the field of psychology. So how do scientists study the brain? So thinking about like, what are these tools of discovery? Early on, as I said, you had like phrenology. There were no tools to map the brain. Today we have lots of techniques. And we want to think about these 2 concepts here, lesion and stimulation. Brain lesions are tissue being destroyed and researchers studying the impact on functioning. Whereas stimulation would be brain regions being stimulated electrically, chemically, or magnetically, and researchers study the impact on functioning. So in addition to sort of studying the brain by analyzing single cell activity, which is kind of like watching a movie 1 pixel at a time, neuroscientists can observe the whole brain in action now. Even as you're sitting there, your mental activity is emitting, sort of signals, whether they're electrical, metabolic, or magnetic, and these would enable a neuroscientist to observe your brain at work. So some of these are EEG, which is a recording of the waves of electrical activity across the brain's surface. And it's measured by electrodes being placed on the scalp. You may have seen some examples of EEGs in movies. So researchers record the brain waves through a shower cap-like hat that's filled with electrodes covered with a conductive gel. Studying an EEG of the brain's activities, like sort of studying like a car engine. You're kind of listening to the hum, but you're not directly accessing the brain. So researchers present a stimulus repeatedly and have a computer filter out brain activity unrelated to the stimulus. What remains then is the electrical wave evoked by the stimulus. A second brain imaging technique, MEG, or magnetoencephalography, measures magnetic fields from the brain's natural electrical activity. So to isolate the brain's magnetic fields, researchers create special rooms that cancel out other magnetic signals, like the Earth's magnetic field. People that would be undergoing this type of neuroimaging would sit underneath a head coil that sort of looks like a salon hairdryer, and participants would be completing activities, and tens of thousands of neurons create electrical pulses, which in turn create magnetic fields. And the speed and strength of the magnetic fields enable researchers to understand how certain tasks then influence brain activity. In CT or CAT scans, X-ray photographs taken from different angles are then combined by computer to show a slice of the brain structure. It can show structural damage. How about PET, MRI, and fMRI scan? How imaging the brain? What are they like? Well, PET scans, positive emission tomography scans, depict brain's activity by showing each brain area's consumption of its chemical fuel, the sugar glucose. So it's a visual display of brain activity that detects where a radioactive glucose goes while the brain performs a given task. And it just shows activity. So how it would work is after a person receives temporarily radioactive glucose, the PET scan tracks like the gamma rays released as a task is performed. An MRI, magnetic resonance imaging, within an MRI, and those are the ones that you're going to go into this almost like tube. I've been in one, into this tube-like structure, and it can be a struggle for those of us that have a little bit of claustrophobia, which I definitely have. You go into a tube-like structure, and the MRI, and I actually had an fMRI, which is what we're going to talk about next. But so in MRI, the person's head is put in a strong magnetic field, which aligns the spinning atoms of brain molecules. Then a radioactive pulse momentarily disorients the atoms. This is a little bit complicated. And when the atoms return to their normal spin, they emit signals that provide a detailed picture of the soft tissues within the brain. Okay. So some really interesting things have been found using MRIs, things like people with perfect pitch have larger than average neural areas in the left hemisphere. And they also have revealed enlarged ventricles for certain disorders like schizophrenia, which is fascinating to think about. And the last one on here is functional magnetic resonance imaging, which is specifically what I had. I was in the MRI machine doing fMRI when I worked in a neuroscience lab at Georgetown University. fMRI is sort of a special application of MRI. It's a measure of blood flow and brain activity by comparing successive MRI scans to show brain function rather than just structure like MRI, showing function as well as structure, and it shows any sort of damage. Where the brain is especially active, blood goes. So these are some of the really interesting studies that have come out regarding many different things, but one that I was a part of was when reading in individuals that had dyslexia, sort of seeing where their brain lit up in the fMRI as compared to normal controls that didn't have reading difficulties. And some of the really fascinating research in that area with dyslexia and fMRI shows that yes, there are different areas that light up for individuals with dyslexia, but with really research-based, high-quality, intensive interventions in reading, that can actually change the areas that light up in people that were previously seen as having dyslexia. The brain areas can look more like the normal controls. So that's really fascinating stuff. So which imaging techniques show structure and which indicate function? So the CAT scans, MRIs, and fMRIs can show structure, and the EEG, MEG, PET, and fMRI can show function. So not just the structure, they can actually show function as well. This is an overview of the various imaging techniques. So I'm going to leave this up here for a minute or so if you want to screenshot this or anything, because this is important to understand the differences between these various brain imaging techniques. Between the EEG, where electrodes are placed on the scalp, the MEG, where you have a head coil, computerized tomography, which is x-rays of the head, generating images, PET scans, which are tracking that temporarily radioactive form of glucose, versus MRIs, which are looking at the structure, where people sit or lie in a chamber. That's what I was telling you about that I've been in. And fMRIs, that are measuring blood flow to regions by comparing continuous MRI scans. And there's some different examples that you can read through on here as well. Okay, so as I just said, that last slide was quite important, as you are going to go on and take the AP exam in psychology. Knowing which brain imaging techniques would be used to show activity or structure is often tested on the AP exam. So pay close attention to that previous chart. Like I said, you may want to actually screenshot it to make sure you're understanding it. And just important to understand that researchers are seeking to go beyond how the brain regions work independently. They're trying to understand the connectivity, right? I do want to pause here for a second before we move on to discussing some of the older structures of the brain and stuff like that, that it's important to realize that there's a lot of neuroscience research coming out that can be thought of as sort of neurobunk. Anything with a brain or brain images seems to be like hot news. And even there have been studies that have shown that even putting like a brain on a picture of a slide or in an article makes people believe that the article is more true as compared to the same article that didn't have the brain image on it. So we just have to be careful of this possibility of neurobunk. And there's actually a really good TED Ed called Beware of Neurobunk that was TED Talk, was created by a neuroscientist who said we should really be skeptical about a lot of the neuroscience research that's out there. A lot of it's still in its infancy. And what happens is the neuroscientists are doing great research and coming up with little tiny, pieces of information, and the media is taking off and utilizing those, over exaggeration of these small little claims, basically. So just be wary, be skeptical, be cautious against overblown claims from the field of neuroscience at this point. So what would you answer? A little comprehension check. Which of the following scanning techniques measures glucose consumption as an indicator of brain activity? So the answer is D, PET scans. Okay, so now we're going to switch a little bit to the older structures of the brain. Okay, and that is the brainstem is the oldest and innermost region of the brain. At the base of the brainstem is the medulla. Here is where you'll find the thing what controls your heartbeat and breathing. Just above the medulla sits the pons, which helps coordinate movements and control sleep. And then the brainstem, including the pons and medulla, you should be thinking about the brainstem, including the pons and medulla, as an extension of the spinal cord. And the thalamus is attached to the top of the brainstem, and the reticular formation passes through both structures. So what are the functions of that medulla right there on the bottom? At the base of the brainstem, as I said, it controls heartbeat and breathing. So it's really important. basic, simplistic things we need to live. Above the medulla is the pons, just above the medulla, which controls sleep and helps coordinate movements. And then there's the reticular formation. It's inside the brainstem. It looks a bit like folded fingers and relays incoming stimuli to other areas of the brain. It also regulates autonomic functions such as arousal. So again, inside that brainstem, between your ears, lies the reticular net-like formation. It's a neuron network extending from the spinal cord right up through the thalamus. The reticular formation controls arousal. Interesting stuff, like electrically stimulating a sleeping cat's reticular formation almost instantly produces an awake alert animal. So it helps control arousal and filters out incoming sensory stimuli. What are the functions of the thalamus? Okay, so at the top of the brainstem is the thalamus. And the thalamus is a relay station for incoming and outgoing sensory information, with the exception of smell. So it sits atop the brainstem. It's sort of a pair of egg-shaped structures that act as the brain's sensory control center. It receives information from all the senses except smell and routes that information to higher brain regions. It also receives some of the higher brain's replies, which then directs to the medulla and to the cerebellum. So what would you answer? Damage to which of the following puts a person's life in the most danger because it may cause breathing to stop? So which one of those things that we just mentioned, that I just mentioned? It would be C, the medulla. Okay, what are the functions of the cerebellum? So you can see where, you see that picture of the cerebellum and where it kind of is compared to the spinal cord. So it's at the rear of the brainstem. And it's sort of baseball size, and the word cerebellum actually means little brain, which is what its two little wrinkled halves kind of resemble. The cerebellum enables nonverbal learning and skill memory. It also helps with things like judging time, modulating emotions, discriminating sounds and textures. It also coordinates voluntary movement. So it processes sensory input, coordinates movement and balance, and responsible for nonverbal learning and memory. So which structures make up the limbic system? So the limbic system is linked to, it's an interesting area of the brain, it's linked to emotions, memory, and drives. And a little way to remember the limbic system is to remember AHH, okay? AHH, amygdala, hippocampus, and hypothalamus. It's located below the cerebral hemispheres. So the amygdala, what are the functions? What are the functions of the amygdala? And the amygdala are, this says lima beans, but I've always thought about it as more like almond-shaped clusters that are linked to fear and aggression. You can see them right there. There's been some interesting research on the functions of the amygdala. One study showed that math anxiety is associated with hyperactivity in the right amygdala. Another study is linked to criminal behavior with amygdala dysfunction. And then finally, researchers found when people view angry and happy faces, only the angry ones actually increase activity in the amygdala. Fascinatingly, in 1939, some psychologists, a psychologist and a neurosurgeon actually removed a rhesus monkey's amygdala and turned the normally ill-tempered animal into a mellow creature. In studies with other wild animals like wolverines and wild rats, researchers have also noted the same effect. And also with human patients, those with amygdala lesions often display reduced arousal to fear and anger arousing stimuli. So how about the other H? What is that one for? That's the first H, actually. The first of the two H's is the hypothalamus, which hypo just means below the thalamus. It directs eating, drinking, and body temperature. It's related to those basic maintenance, eating, drinking, body temperature, and even sex drive. Helps control the endocrine system and governs the pituitary gland. So what does research suggest about the functions of the hypothalamus? Interesting stuff. A really interesting discovery happened when two neuropsychologists were trying to implant an electrode in a rat's reticular formation, and they made a mistake. They placed it incorrectly, and weirdly, as if it was wanted more stimulation, the rat kept returning to the location where it had been stimulated, and by the misplaced electrode. And when the researchers realized that they had placed it in the hypothalamus, they realized they sort of stumbled upon a brain center that provides pleasurable rewards. So as this picture shows here, stimulating the reward center of the hypothalamus will actually motivate a rat to cross an electrified grid, receiving painful electric shocks to reach a bar that will give additional stimulation to the hypothalamus. How about the other H? There's the other H, the hippocampus. And a way to remember this is sort of think about hippos have a good memory on campus, hippocampus. Because the hippocampus is linked to conscious memory. It's sort of a seahorse-shaped brain structure that processes conscious, explicit memories and decreases in size and function as we grow older, sadly. Humans who lose their hippocampus to surgery or injury also lose their ability to form new memories or facts and events. And there are some really famous cases of case studies within psychology about patients who have lost their hippocampus or have had it damaged significantly. So what does research suggest about the function of the hippocampus? Humans who lose their hippocampus to surgery or injury lose their ability to form new memories. That's very fascinating. Those who survive a hippocampal brain tumor in childhood struggle to remember new information in adulthood. And finally, NFL players who experience loss of consciousness, concussions, one or more of them actually, may later have a shrunken hippocampus and poorer memory. So what would you answer? Damage to which of the following brain structures would affect the processing of new explicit memories? Remember, what was that animal that had good memory on campus? The answer is actually D, hippocampus. Okay, and we're back to our learning target reviews. Let's see what we know. Can we describe several techniques for studying the brain's connections to behavior and mind? So case studies and lesioning first revealed the general effects of brain damage. CT and MRI scans can show us anatomy. EEG, MEG, PET, and fMRI recordings can reveal brain functioning. How about describing the components of the brainstem and summarizing the functions of the brainstem, thalamus, reticular formation, and cerebellum? So the brainstem, automatic survival functions is what you should be thinking when you hear about brainstem. The medulla is what we need to basically have the basic components of life, heartbeat and breathing. The pons, just above that, coordinates movements. The reticular formation filters incoming stimuli and affects arousal. The thalamus is our sensory control center. And the cerebellum is involved in coordination and balance and processing sensory information. Finally, the limbic system, we have to describe the limbic system. So the limbic system is sort of involved in emotions, memory, and drives. And it's comprised of three parts, the AHH, the amygdala, which is involved in aggression and fear, the hypothalamus, which is utilized for maintenance functions, sort of pleasurable rewards and control of the endocrine system, and then the hippocampus is responsible for conscious memories. The pituitary gland, the master gland, controls the hypothalamus by stimulating it to trigger the release of emotions. So that is it for module number 11. Thank you for listening and take care.